Hello students, welcome to LEELA'S tutorial. So today we'll start with the applications of plant biotechnology. So let us see the topic, the meristem culture and production of disease-free plants. What do you mean by meristem? Meristem is the fastest growing part of the plant body. It can be apical meristem or it can be an axillary meristem. Or you can say it can be a shoot meristem or root meristem. So if you take a meristem, it can be in the shoot or it can be in the root tip. Right? Apical meristem means if it is at the tip of the plant, you call this as apical meristem. Right? If it is at the axil of the leaf, then you call it as axillary meristem. Or if it is at the tip of the root, then you call it as root tip meristem. So here you can make use of any meristem in order to produce the disease-free plants. Right? It can be any microorganism or any other parasites or pathogens or virus. So let us see. The application is to eliminate the virus and other parasites by culturing the plants in vitro. Right? So if you take a complete plant, see here I have taken the plant which is having viral infection. That means if I take a fruit, flower or any other part of the plant, what will be there? The viral infection will be present. Right? So if I take the seeds and sow it again, the plant which is grown or after germination, it will also have that virus. So I have to produce a plant in such a way that I must get rid of these pathogens or the parasites. So here let us see the steps involved in it. The first step is initiation of the callus. So here I told, though I take any part of the plant, the viral infection will be there. So why I am taking only the meristem? Right? So let us see the structure of the meristem. This is the apical meristem. Right? So when you zoom it, it is this one. Right? The apical meristem which is present at the apex of the shoot. Right? Axillary meristem, the meristem which is present at the axil of the leaf. Okay? So if you take a meristem, here you have the younger leaves. Right? You have the younger leaves. And this is the apical part, the tip, the complete tip of the plant which is responsible for growing upwards. Right? The length of the plant is increased. If you take the root meristem, the length of the root will be increased. Right? So this is the meristematic cells which are highly proliferating. They divide rapidly at very faster rate. Right? So here now this meristem is protected by primary leaf primordia, then secondary leaf primordia, tertiary leaf primordia, and gradually they transform into younger leaves. As the meristem is growing, these primordia will develop as an younger leaves. Right? So and meristem will be growing and increasing the length of the shoot. Okay, so now we are taking only this part. If I take any other part like leaf or shoot or axil or nodal or any other region, everywhere or every part consists of this virus. Right? But I want to produce a plant which is virus free. Okay, so what I will do, I will select this meristematic tissue or the apical meristem because the rate of dividing, the proliferation of the meristem is very much high when compared to the spreading or transferring of the virus from one cell to the another cell. Right? So I told if there is a viral infection, the complete plant will have this virus. So here it is just now it is dividing. Right? It is dividing at a very faster rate. By the time this virus divide and get transferred from one cell to the another, it may not reach this apical meristem. That is the reason we are making use of meristem or apical meristem as explant. Is that clear? Explant means any part of the plant body which can be used to produce a complete plantlet. But here we are making use of only the apical meristem so that there will not be any virus. The virus may be dividing and spreading to the younger leaf primordia, right, but not to this meristem, because by the time it reaches to the apical meristem, the meristem will be growing in size, right. So there will not be any contamination in this particular region. Okay, so let us go into the procedure. So the steps involved in meristem culture are initiation of culture, callus. That means when you introduce this apical meristem, either you produce the callus or you directly go for the shoot formation. Okay, so second one is shoot multiplication, root development after the shoot is produced, developed. Then last one is transfer the plantlets to the soil so that they get adjusted to the new environment, because till now you are growing in plant tissue culture media, right. So here this meristem culture is also referred as shoot tip culture or tip culture or culture of shoot apices. Is that clear? So then here first step, you are going to select a diseased plant. So now from this diseased plant, we want only the apical meristem to be taken. It can be apical or axillary meristem. Okay, I am taking the apical meristem. So I trim the shoot. And I want only the shoot tip now. Okay, so here meristem is present. Primary leaf primordia, secondary leaf primordia, tertiary leaf primordia, then younger leaves. Okay, this complete structure. So after taking this, I have to go for surface sterilization. Surface sterilization is done by using mercuric chloride, sodium hypochlorite, right. So here by surface sterilizing, you are removing the contaminants or the dust particles which are present on the surface of the explant, right. So after surface sterilization, you are taking it to the laminar airflow in a petri plate placed with tissue papers, right. Allow it to dry. Then with the help of scalpel, you are going to trim only the apical meristem, right. As we told, there may be viral contamination here, right. So you are going to trim only the apical meristem. If the meristem size is very much small, then you are going to take even the primary leaf primordia. If the meristem size is much more big, you no need of taking those leaf primordia. You can trim even those. Okay, so normally we take 0.5 to 1 mm length of this meristem. After trimming this apical meristem, you are going to introduce that or inoculate that in plant tissue culture media. So after inoculation of the apical meristem, it is the incubation. So when you incubate at favorable temperature, right, with light, okay, temperature, light and also humidity, what happens here, gradually due to the plant growth hormones you have added, there will be shoot formation. So once you get the shoot formation, here the shoots are transferred into the rooting media. Okay, then the root formation is observed. Then after the root formation, when you get a complete plantlet, you are going to transfer that into the soil. So in between you have the greenhouse where the acclimatization or the hardening process will be done, so that plant will get adjusted to the new environment, because till now you are growing in ready-made media. Now the plant has to survive or fix it in the soil, in the natural environment. Then you are allowing them to grow in greenhouse, okay, where humidity, light, temperature, everything is maintained. Then you are transferring them to the outside environment. Is that clear? So now these plantlets which are produced are free from virus, and they are called as virus free plants, and these plants can be given to the farmers so that they can grow in their fields, right, so that they get virus free or disease free plants for their harvesting or cultivation process. Is that clear? If you have any doubts, put it in comments.